This video introduces our graphical user interface developed as part of the paper, a visualization framework for localized surface plasmon resonance imaging in sensing applications. Localized surface plasmon resonance imaging is a powerful technique used in clinical diagnostics and environmental monitoring to detect various molecules simultaneously. The imaging process uses multiple narrowband light sources to scan a sensor composed of tens or hundreds of plasmonic nanostructure spots, each functionalized to bind to specific molecules. A monochrome camera captures the transmitted and incident light at each spot, across different wavelengths and time steps. To detect molecular interactions over time, the first step is to identify the spot locations. For each spot, a central area and a surrounding ring are extracted. These represent the transmitted and incident light. From these, we compute the extinction value, which reflects the combined effects of scattering and absorption. For each spot and time step, an extinction curve is generated by plotting extinction values across wavelengths. By comparing these curves at different time points, we can identify changes that indicate molecular interactions. Traditionally, domain experts perform much of this analysis manually, often using custom scripts. This process can be time-consuming and makes it difficult to experiment with different parameters. Our framework integrates the entire analysis pipeline, automating key steps while giving users full control over configurable parameters. This flexibility is essential, as many analysis choices remain underexplored. The tool is organized into three main tabs. The first is the Spot Detection tab. Once a dataset is loaded, a representative image is shown in the main view. Spots are detected using the Hauff transform, guided by user-specified parameters like approximate spot size and spacing. These can be input directly or measured using tools built into the interface. After automatic detection, users can fine-tune the results, adjusting spot positions, radii, or adding and removing spots as needed. Next, spots can be selected and grouped for analysis. Grouping is useful when multiple spots are functionalized to detect the same molecule, improving the signal-to-noise ratio. Finally, the regions for incident and transmitted light are defined. A histogram of pixel intensities helps users assess whether their selection is appropriate. The second tab provides the core analysis features. Here, users can view extinction curves for each spot and time step. If spots have been grouped, curves can be averaged, either by combining the curve's metrics or by averaging the underlying extinction data. To reduce visual clutter, curves can also be averaged across time ranges. These are shown with dashed and solid lines, representing different time intervals. This averaging is for visualization only and doesn't affect the underlying computations. Users may also choose to display raw data alongside calculated metrics. The sensorgram view shows how selected metrics change over time. Users can define several time ranges and compare the results in the statistics view. The sidebar provides additional controls to test different parameter settings or model choices, including regression methods or algorithms for calculating the curve's characteristic wavelengths. The third tab allows for detailed metric inspection per spot. For example, one view shows the absolute difference in mean wavelength between two selected time ranges. Spots with detectable changes are highlighted with a black ring. Users can explore different metrics, color maps, and transparency settings to customize the visualization. In summary, our framework brings together all steps in the localized surface plasmon resonance imaging analysis workflow, making it more efficient, flexible, and user-friendly for researchers.